Hello and welcome. I am an academic clinician working in London and Oxford, and today I would like to tell you about how I've been working to translate microbial genomics for public health benefit using the MenDeVar index. This work started over 5 years ago when the 4C-MenB vaccine was introduced into the UK infant immunisation schedule, with outstanding questions about the genetic diversity of the proteins contained in the vaccine, methods by which to analyse these, and the relevance and implications of this diversity. In 2016, we published a curated scheme for bacterial antigens established within PubMLST. Whole genome sequence data allowed identification of deduced peptide sequences for each vaccine antigen, and careful curation allowed characterization of the diversity of antigens in both MenB vaccines, with bacterial antigens listed here and with member containing FHBP. This information is available on the isolate record and amongst the typing schemes. However, it also became apparent that for any given MenB isolate, the impact of these two licensed vaccines could be different, and it was therefore important for healthcare professionals to know this and be able to assess their isolates. Genotypic typing we developed, such as the BAS typing scheme or FHBP peptide typing, provided only presence-absence data and no information about protein expression or cross-reactivity. But at the current time, there are only limited laboratory assays available to do this, and they're not universally accessible. We decided to develop the MenDeVar index, complexing genotypic information, the vaccine antigenic variants, and phenotypic data from laboratory assays that were developed to assess cross-reactivity, protein expression, and bactericidal killing. For each antigen, each antigenic variant was assessed, and the phenotypic data was assessed. For Bexero, it was for MATS assay data, and for Tremember, it was measure and SBA data. I'll talk you through this algorithm briefly, but if an antigenic variant had been tested in more than 5 experiments, and it was assessed to have met the inherent assay thresholds being covered or not covered by the vaccine, it was then determined the degree to which these antigens were covered. So if over 3/4 of isolates were deemed covered, then antigens were amber. If over 3/4 of isolates were deemed not covered, the antigen was red. And if any antigen was not tested in more than 5 isolates or had not met the amber or red criteria, it was deemed grey. We then had to assign the index to isolates, but accounting for all their antigens. If an isolate contains at least one of any green or amber antigen, it is labelled as such. If isolates contain all the red antigens, it is labelled red. If it is none of the above, then it's labelled grey. The MenDeVar index was developed to combine the complex genotypic and phenotypic information, but also to translate this into a simple, evidence-based output for healthcare professionals. As many reference laboratories are now sequencing meningococci in real time, this information can be available hours after sequencing. Here we can see how it appears on an isolate record under the secondary metadata heading and vaccines. For each vaccine, Bexero and Tremember, you get your output with both a traffic light and an explanation. We see which antigen has been contributing to this outcome, and we can see the evidence base for both Bexero and Tremember with the assays used and the published data. So working in collaboration with the Scottish Microbiology Reference Laboratory and Public Health Scotland, with Andrew Smith and Claire Cameron and Ian Fevers, we collated this data and developed a robust set of rules that we thought would be useful to inform healthcare professionals. And it's been implemented in the NHS in Scotland since 2018. We are in the process of developing a wider oversight committee that will be able to continuously update MenDeVar index and with new published data so it remains an up-to-date evidence base for health professionals. Many colleagues that have been involved in this work and also funding sources, the Wellcome Trust and Thrasher. But now I want to show you how you can access MenDeVar. There are 3 main ways. You can go through the isolate record. So starting here in the PubMLST Neisseria database, you can go to the isolate collection. You can search the database for any given antigen of interest. Sorry, isolate of interest. And you can search on that page and see the data I just showed you, secondary metadata here. And you can also search the typing, Bexero typing. And you'll see not only the antigen profile, but also the Tremember and Bexero reactivity, which you can download. The second way to do this, if you haven't got isolates on the database, but you have sequencing data or PCR product data of your vaccine antigens, is you could search here, the MenDeVar plugin. If you launch MenDeVar, you can then search sequence and instantly get a result showing you which output you have for each of the vaccines, along with the same information, which peptide and the evidence base. There are also additional caveats and information that are here and also found on the isolate record. The third way you can do this is if you already have a list of isolates and you already have all their best profiles, is that you can search via the typing section and type in a batch. You can search by bacterial antigen sequence type and paste your profiles in. You'll then get an output of all your 20 isolates that you searched on the profiles and the reactivity for both vaccines. We hope that you find this useful and we'll always welcome any feedback. Thank you very much.